Hi everyone, thank you for coming to today's talk. I'm a grad student in Professor Rajapakse and Lindsay Moyer's lab, and today I'll be basically be going over three different tools that we built over the past year, um, focusing more on like going from biology as a uh, experimental versus trial and error phase to more computational side. With that, if you guys have any questions, just feel free to stop me in the middle and put forward your question. And with that, I will get into it. So briefly, I'll just go over the core biological background and the math background required to follow the tools that we built. And then I'll go over what the hardwired genome is, how we constructed this hardwired genome purely from data. And I'll show some interesting applications that we did with this hardwired genome. And I'll also go over what the future holds for digital biology in a bit. I also worked on a specific file format that is memory optimized. So I'll go over what challenges we face when coming up with the idea and why we should not have too many file formats, especially in biology. And I'll go over a automated agentic system that we built to like identify and generate biomarker from data. Okay, uh, to go the background. So each cell, like we all know that every cell has the same genome, but how do cells differentiate into like a fixed number of 300 different cell types? So each of these cell type is sort of, you can sort of think of it as a stable basin, and the cell as the marble rolling down this hill with different diverse um, diverse decision points. And at each of this decision point, the cell is pushed towards one or the other side. And as the cell moves down the hill, it gets more and more differentiated into these different cell types. This idea of like a physical marble rolling down was ideated by Waddington, and this is the Waddington landscape the famous Waddington landscape. Um, and as the cell goes down, it gets more specificity towards that particular cell type. How do each of these decision points, like at each of this point, how does the cell decide which direction to go to it? This is mainly guided by these certain proteins called transcription factors. So there are these proteins that bind to specific parts of the DNA, and then it promotes the transcription of a specific gene. And there are different categories of transcription factors. There are transcription factors that specifically affect a single gene, and there are transcription factors that can affect entire networks. They can bind to multiple different genes or like entire regulatory networks called master regulators. And there are normal functional proteins which genes code for, and these proteins sort of give the cell a specific phenotype. For example, in a specific cell type, it gives the specific cell type the cell type specific characters. In 1989, Harold Weintraub, purely through trial and error, figured out that there are certain transcription factors which can move a cell towards a different cell type. So you can sort of think of this as we have human fibroblast here, and Harold Weintraub found out that if you add MyoD to human fibroblast, you can move it from this stable state to a different stable state. Here we get like from fibroblast to skeletal muscle like cells. And in 2007, Shinya Yamanaka showed that with a cocktail of these four transcription factors, OX4, SOX2, famously called the Yamanaka factors, if you add it to a human fibroblast, you can move the cell up, like we can roll the cell towards the top of the hill and make it more pluripotent. In essence, we can take a differentiated cell and make it more pluripotent so that it can go to different cell states. This is extremely interesting because what we once thought of as like states fixed, we can now we can sort of essentially move it back, and the applications of these are extremely enormous. We can do a lot of things with just by reprogramming one cell to other. And this method of moving one cell from one state to the other is called cellular reprogramming. But the interesting part is both of these was purely based on trial and error. Like both of these extremely great scientists, they just tried out each transcription factor, see how it changes, and then decide which one works and which one doesn't. We sort of want to move towards a computational field, like with the amount of data that we generate and the computational capacity that we have, we sort of want to move it towards a more deterministic field. Now, here we can frame this particular reprogramming problem can be framed exactly as a control value. What do I mean by that? So we have this famous classic control equation dx over dt equal to ax plus bu. Here, I'll explain what each of these parts are. So dx over dt is how x is basically the expression or any variable that we measure from a particular state. Dx over dt tells us how it changes over time. X of t is the measurement, and A is sort of like a state transition matrix where we know how from the previous time point to the next time point, how the system will move, given there is no input. B is the additional input that we give to the system externally. And U is the external input, actual input that we give to the system. And these two, like the natural dynamics and the input determine how the system moves over time. And in 2017, the famous hardwired genome was coined by Professors Mail and Professor Rajpakse. What is hardwiring the human genome? So we have the genome, and on the genome we have certain set of genes that are that code for certain proteins. Now here we can see there are three, as I discussed, there are three different categories of transcription factors or three different categories of protein. Here gene A transcribes for protein A, which can then localize back to the nucleus, and then it can regulate itself. So this gene codes for a protein that can regulate itself. Similarly, gene B codes for a protein B, and this protein does not regulate itself, but it can go and regulate other genes or other transcription factors in this case. And gene C is just a normal gene. It codes for a particular protein. This protein does not localize to the nucleus, but it goes and interacts with other proteins and sort of gives rise to specific function at the end of the translation. So this is what this essentially tells us is we have the genome. Every gene is hard coded or hardwired within the genome. But how these interact with each other, that is also hardwired. And these interactions give rise to different functional outcomes. We can define this as a linear algebraic way. So here we see this is the hardwired genome, which basically tells us what each of these genes are and how the genes interact with each other. And we have the B matrix. This is exactly similar to the control matrix that we had in the previous slide. And the C matrix, which is basically how transcription factors interact with other transcription factors. Moving along, I'll give a basic introduction to network theory, because this is the core of what we're going to build on. A graph is essentially a set of nodes and a set of edges on how each of these nodes are connected with each other. So here we can see that there are 4 vertices and there are 4 edges that connect the different nodes. The thing to note here is that not every node is connected to every other node. We have interactions between the 4th node and 2nd node, but the 1st node and 4th node don't interact with each other. If everything interacted with each other, then that's essentially inherently that's no meaning in it. And this network, this graph can also be represented as a linear algebraic matrix that we discussed earlier. And here we can see each of these rows are nodes, and each of these columns are also nodes. So we have nodes by nodes, and wherever you see one, it's an interaction between the node in the row and the node in the column. So here we have node 1 interacting with node 2, which is this edge, and here we have node 2 interacting with node 3, which is what this edge is, and so on. Here we only have ones and zeros. The interaction is binary, which means there is no weight to how much these two interact with each other. And we can see that the matrix is symmetric, which means so everything on top of the diagonal is symmetric to everything below the diagonal, which means there is no direction to the particular network. Like V4 interacting with V2 and V2 interacting with V4 are exactly the same. Now that we have a graph, you can take this graph and attach some biological context to it, converting it into a network. Here, from now on, whenever I talk about nodes, it represents a gene, and the edges represent the molecular interactions between those two genes. These molecular interactions can be determined purely from data. I'll explain how we do that in the upcoming slide. This you see here, whatever you see here is a huge A matrix that we defined before. Before we only had 4 nodes, here we have 20,000 genes by 20,000 genes. And this is just a preview of the tool that we built. I'll show a quick demo at the end of the presentation, but we can see that this is just a montage of how each of these genes interact with other genes. Now that we have all the baseline covered, I'll explain how we extract this particular hardwired genome from data. So the data that we use comes from 2 databases called Curie and String. These are both protein-protein interaction databases, and they use specific methods to validate this particular interaction. So Curie uses E2 hybrid string, and String is basically like they use like functional validation to extract the protein-protein interactions. And then String also gives a sort of confidence score, which ranges from 0 to 999. And as I said, if every node interacted with every other node, there's no inherent meaning to it. So we sort of threshold this particular confidence score to get a like sparser network, which contains richer information. And from this, proteins, as I mentioned, proteins are a byproduct of transcription and translation, which is coded by the gene. So proteins can be thought of as like a proxy to actual gene-gene interaction. So from this protein-protein interaction database, we extract a set of genes that interact with each other, and we convert this into the A matrix that we have. The A matrix now contains genes by genes, and every black dot that you see is a 1, and every white you see is 0, exactly the same as the adjacency matrix that we saw. Going on, now we have this huge matrix. So this matrix, we still haven't talked about specific cells. This matrix comes from all different cell types and interactions from all the samples that we have. We don't go into a single specific cell type. So we need to define what from this matrix, how can we extract a particular cell type? And this is just a cartoon explaining how we define these cell types. So here we can see there are 7 nodes and 7 edges, 8 edges, I think. So this edge configuration leads to this adjacency matrix, and this adjacency matrix is sort of like a linear representation of cell type A. So this gene interaction network gives rise to cell type A. And similarly, this configuration B has certain edges. If you notice, certain edges are gone, but certain new gene interactions are there. And that gives rise to this adjacency matrix, which is also symmetric and square, since we are unweighted and undirected. And this gives rise to a different cell type B. Okay, now we know now we know how we can define cell types. How do we extract these cell types from data? So we take bulk RNA sequencing data for a particular cell type. And what we can do is we sort of do naive binary thresholding at we can do mean or median based thresholding. And what we get is every gene that is expressed higher than that particular thresholded value, we consider it as active in that particular cell type. And if a gene is below that particular threshold, we consider it as inactive in that particular cell type. And every red line that you see here is an active gene. And everything that is not coded in red is inactive. So you can sort of think of it as we are extracting these nodes from the original hardwired genome to get a smaller matrix, which is also gene by gene, but these genes are expressed in a particular cell type. So we get the interactions for a particular cell type within here. So here we can see the network for myotube, and here we see the network for fibroblasts. Why we chose these two, I'll go further and I'll explain. Now we have the cell type specific matrices. We need to extract the B matrix, the control matrix that we discussed initially. So how do you extract the control matrix? We need to determine which of the genes that we have in the entire A matrix actually are transcription factors. So we took a list of 1600 about 1600 transcription factors from this the human transcription factors, which gives us this list. And from this list, what we can do is we can extract similarly in a similar way to how we extracted the cell type specific network. We extract just the columns that correspond to this transcription factor. Why do we need only the columns? This is because the B matrix sort of tells us how a transcription factor affects every gene in the system. So now we were dealing with genes by genes in the A matrix, but once we extract the B matrix, we are dealing with transcription factor and how they affect the gene. So you can think of it as if given a particular transcription factor, we can know which of all the genes that it has an interaction with and how it will go and affect all of these. And going one step further, we can also, the same list that we had, that we extracted the columns from, we can also extract the rows from this B matrix to get a sense of how each transcription factor interacts with the other transcription factor. So we get a transcription factor by transcription factor matrix, which gives like a overview of how each of these networks are constructed, like these gene regulatory network. We know how, like it's like a domino, how one introducing one transcription factor can affect other transcription factors. And from this B matrix, what we did is we were interested in the original transcription factor that Harold Weintraub used to convert a fibroblast to a skeletal muscle, and he used this transcription factor called MyoD. Now, MyoD is sort of like a master regulator that regulates all the other transcription factors and genes that are responsible for this myogenic lineage, which is the conversion of fibroblast to myotube. And what we essentially have here is we have a column and we have a row. The column tells us all the genes that MyoD affects. Like MyoD has interactions with all of these genes, and the row tells us which transcription factors are regulating MyoD. Now, I'll reiterate this row. This column gives us all the genes that MyoD, the transcription factor, affects, and the row gives us all the transcription factors that affect MyoD. So we can think of this as if we go to for example, if we take a fibroblast cell type, and we can see out of these transcription factors that actually affect MyoD, which ones are actually crucial for that particular cell type. We'll be able to distinguish those, and then we'll be able to sort of infer that from this particular information that we get. Now, Now that we have the control matrix, we have the start state and the end state, we need to we sort of need to push one cell type to the other cell type. That's basically what we program. Now, what we see here is the same configuration that we had previously, and we add a specific transcription factor. We introduce this transcription factor into a network. Now, this can be thought of as, in an experimental sense, this can be thought of as adding mRNA into a cell and pushing that cell towards a specific target, or like doing extra edits to increase transcription of a particular transcription factor. And what we see here is this transcription factor. We know all of its interactions from the previous B matrix. So we know all the interactions that it has and all the other transcription factors that act on it. So what we can essentially do is we can add this transcription factor from the original full hardwired genome. We know all of the edges that it contains. And what we can do is, if we take a look at this G1, what happens is G1 will have certain edges. And once this transcription factor acts on that particular gene, it can either be activated or repressed. So this classification of transcription factors is I think I skipped this a little bit. So the transcription factor, when it binds to a particular gene, it can either increase or decrease the transcription. So this distinction is extremely important, as if this transcription factor acts as an activator, then nodes that don't exist will get activated, and they'll form edges with existing nodes. And if it acts as a repressor, for example, it will remove the nodes from a particular vertex to particular gene, and then reduce the expression of it. So the protein-protein interaction doesn't happen anymore. Does anyone have any questions till now? So what we essentially did is we took this particular framework, we took fibroblast specific A matrix that we had, we took the transcription factor as MyoD, and what we did is we sort of constructed this as an algorithm where you can add MyoD to this network. You can see how it affects the edge structure of all the other nodes, and we can see that there are certain genes that have been affected, and they have their nodes changed. So now, if you can think of this as this edge didn't exist, but now it exists. So this means if this is a transcription factor, then the expression of further genes will also get affected. So further genes will get activated, further genes will get repressed, and so on. So you can think of this as imposing in experimental terms, we add MyoD, and then after a while it goes and attaches to all the first order interactions, and let's say those transcription factors further go along the genome, and they affect other transcription factors, increasing sort of like the domino. So what we did is we constructed this as an algorithm, and this algorithm basically goes through the specific cell type that we have, and based on the activity of the transcription factor that we insert into the network, it can go and change the structure of all the genes that it interacts with in the first order, and then we can also go further, like second order, third order interaction. Now, why is this exciting? We saw that we framed this as a control problem, and now we have a way of basically validating if the control problem that we stated, the outputs that we get, like the optimal inputs that we get, we can sort of validate in silico without having to go and perform actual experiments. We can filter out to sort of fine tune these recipes and then make these exact better recipes without having to go and perform the experiment. And that's the core idea. But here we have a slight, here we are stuck in a way. Why is that so? Now I mentioned that a transcription factor can act as an activator or a repressor. And there are 1600 transcription factors. But out of these 1600 transcription factors, we only know the activity of about 50% of those. So we have about 174 activators, 705 repressors, a lot of repressors. But 729 of those 1600, we still haven't classified as activator or a repressor. There are slight nuances to this. Why can't we? Why is this a hard problem? Why is this a challenge? Essentially, transcription factors can act as activator or repressor depending on the specific context that we give it. For example, in a specific cell type, a transcription factor that acts as an activator, in a different cell type, it can act as a repressor. It can essentially perform different actions based on which cell type or which tissue type that we are dealing with. The second part is transcription factors go in and bind to the actual DNA sequence, as I mentioned earlier. Now, in the DNA sequence, which part does it go and attach to? There are multiple places in the genome that the transcription factor can go and attach to. For example, there are regions called promoters where the transcription factors can go and bind. There are enhancers, but within these enhancers, there are multiple, like you can have overlapping positions, you can have a variety of transcription factors that bind together and then like they have a combinatorial effect. So all of this makes this problem extremely challenging. And the previous algorithm that we described depends extremely on what the activity of the transcription factor is. Now, the B matrix that we had, the original algorithm that I said, we sort of assign a transcription factor a specific activator or activator or repressor. So if we see here, this entire column that we have, MyOD acts as an activator. But as I mentioned right now, that can change depending on the context. So you can have, like, for example, in a certain interaction, it can act as an activator. In a certain interaction, it can act as a repressor. So we sort of need to determine this B matrix and extract a richer B matrix where we get ideally all the transcription factors, all the genes, and exactly how they interact. So we get this entire matrix of activity, activator, repressor, and we get multiple B matrices for each of these cell types. This is cell type specific. So we sort of need to define this, and we need to understand how this works. So with recent, this is a quote from Professor Cook from Oxford University. So he's basically saying in the last year, the evidence is evidence suggests that every transcription factor can act as both activator or a repressor, and every motif can also be activating or repressing. And this is extremely exciting because this is like a very unexplored and challenging but very interesting problem that can directly affect or directly help us in the reprogramming problem. So in the recent years, there have been a bunch of AI-based sequence to expression models, such as Alpha Genome. There's also, I think, a lot of these models exist. So what these models claim is, given just a DNA sequence, the model has learned how each of these nucleotides interact with each other and how in a specific cell type, in a specific tissue, how this particular genome structure can sort of express genes. So you can sort of predict how these expressions change based on the DNA. Now, what we want to do is, there are within the sequence, we can sort of get these motifs where transcription factors can go in and bind. And our goal with this particular classification is to sort of use the DNA sequence. We have this DNA sequence. We change the transcription binding site, like the binding motif, to whatever we want, to a specific transcription factors, and then we can sort of like use these existing sequence to prediction model and classify these transcription factors as activators or repressors. And this is currently what I have been focusing on. 800 odd transcription factor information that we already know, and sort of sort of use it to guide these models to classify these as activators or repressors. And I just want to go over this quote by Demis Hassabis. Um, so he's saying, if we biology is not just a static, like it's not just a static thing that determines how every cell will react. But if we want to understand how a cell will behave, we need to understand the dynamics and interaction between the different parts of the cell. So which is this is very interesting, because in this talk he also says that AlphaFold is looking more at just the structure, which is very static. It doesn't include any of the other like dynamics that guide how a cell or how biology actually works. So with that, I'll go over a small demo of the tool that we built from the. Okay, so basically right now it's only deployable locally. Um, you can I'll provide the links to the repository so that you can set it up locally and then do this. So here, what we see here is the actual A matrix, the entire A matrix loaded up, uh, and what we can you can go in and look at certain parts of the A matrix and see which genes interact with each other. So we can take a look at a specific region. So yeah, here I'm just looking at a specific region of the A matrix, and what we have is 500 genes by 500 genes interacting with each other, and we can visualize this network. And here what we see here is the 500 genes ordered based on the centrality scores that they have. So nodes that are in the middle are highly central. So here we see that it has degree 17, and nodes on the outside are less connected nodes. So we have degree 1, degree 3, and so on. Now, why can't we look at the entire 20,000 by 20,000 genes? It's because that's like a extremely huge matrix, and looking at it all at once won't provide you any sort of information within within the computation power that we have. Like we don't want to load up that amount of data into memory. And here we can see all the purple nodes that we see here are master regulators, as defined in the previous slides that we saw, and all the pink nodes are transcription factors that affect other genes, and every light colored cell is a gene, normal gene. And what we can do is uh, this straight away tells us within that particular subsection which genes are important, and those genes show up in the middle. And you can also provide your own custom set of genes. For example, once you threshold a specific cell type, you can look at a cell type specific network. And that right now I have the data for fibroblasts loaded in. So if we look at this fibroblast specific network, I mean, as expected, most of these nodes don't have any interactions. So we can go in and see which, or within the fibroblast network, within those set of genes, which genes are ranking higher based on these. We also have a bunch of other metrics such as eigenvector centrality, which tells us how a gene is affects like global dynamics, and um, normal like other centrality measures. And you can also calculate how out of the genes, if certain genes are responsible for certain like cell cycle related genes and other stuff. And within this network we can add MyOD, and this is the perturbation algorithm that I talked about. And what we've essentially done is we've forcefully added MyOD into this network. And as expected, MyOD has a lot of interactions, and every pink node that you see here is a first order interaction of MyOD. So here we can see that this particular gene is directly affected by MyOD, and a bunch of other genes also get affected because of MyOD. And then we can further propagate this algorithm, and then we can calculate certain measures from the source to target, and we can sort of see how this works. We can also, instead of MyOD, we can add our own transcription factors and find out how they move to the target. Okay, moving on to the second part of my like this is a slight deviation from what we were doing. Now, whatever we've been discussing has just been the function of a cell. So we talked about like gene expression, we talked about how genes interact with each other. All of this leads to a specific function, but that's just one half of the story. We also need to understand how the genome is folded within this nucleus. That in turn guides how the transcription factors actually affect. So if certain parts of the genome are close together, those get expressed together, sort of like transcription clusters. In order to do that, we need to perform some other other techniques. I'll go with them. But in general, I want the problem that I was specifically interested in is there are a lot of file formats specifically to store biological data. So here we see this is just a conservative list that I put together based on what I know. I'm pretty sure there are hundreds of other text files with data dumped everywhere. But this this sort of covers a fundamental problem. Like, for example, most of these file formats are also just raw text files, but with a specific structure to them. And this creates a in software and information technology was in a phase at some point. There were like hundreds of extensions. Everything did the same sort of task, but here in biology, this makes like transferring or like scientific dissertation extremely hard. So this interesting paper just tells us how it was in the 1980s and 1990s and how they solved this particular standardization problem. The thing is, there is a fundamental requirement for these different file formats and file extensions. This is because one file format cannot cater to all the problems, and it cannot solve all the it cannot be a solution for every problem that people have. Now I'll just go over HiC and why we need like a structure, why we need to how and why we need to understand structure. Now we have the genome. We can sort of cross-link the genome with formaldehyde, and we can we can connect them and sequence this particular part of the actual DNA. So we know which two parts of the DNA are close together in three-dimensional space. So we sort of get a interaction. Again, we get a adjacency matrix of which part of the gene is closer to which other part of the genome, and then we essentially get this huge A matrix. And this method was popularized by Eris Lieberman Aiden in 2009, and we sort of get the structure within the nucleus. And this data is chromosome. We get the genome-wide coordinates by genome-wide coordinates, which you can think of it as like almost 3 million to like 10 million by 10 million. And this data is extremely huge. We need to find extremely efficient ways to store this. And one project that we were involved in, it required us to look at specific loci, and then the entire interaction profile of that particular loci. So, for example, if we look at a specific locus here, we want to extract the entire interaction profile of that locus without having to deal with all the others of it, because we were only interested in this. One of our postdocs was training a model for HiC, and this was a problem that we were faced with. Now, HiC data is not very straightforward. You have this genome by genome matrix. You can also bin these genome by genome matrices at different resolution. So you can get like a 1 megabase bins and then sum them up together. So you get a smaller matrix. And then you can also do it at multiple different resolution. And what you see here is from a specific tool that we can see these different levels of resolution. And if we think about it, how do we store these huge data matrices and how do we load it up to a computational memory? So at the 1000 base pair resolution, which is the extremely fine resolution, you get a huge amount of bins, and loading up the entire matrix, you will get, if you load up the entire matrix, you get about this number of elements because it's symmetric, and that will require around 8.7 terabytes of memory to load it into your computational memory. Even at 10,000 base pair resolution, you get around 300,000 bins, and loading a 300,000 by 300,000 bins will take around 100 gigabytes to 200 gigabytes of memory. And people have come up with different file formats to store these matrices in an efficient way so that we can load them up without having to deal with the overhead of having huge amounts of memory. So these are a few data formats that we have. For example, cool files are store only the positive interactions and leave out all the zero interactions. So we essentially get a compressed sparse array, and then we can load specific parts of it. And HiC matrices also store it as binned, like at different resolutions, you get a certain sub matrix, and then you can load it into memory without having to worry about loading the entire matrix. But for our use case, like we wanted to fetch random loci, as we said, and none of these formats would be useful for this. For example, in Cool, it's if you want to load like a row in the middle of the matrix, you'll still have to load through the entire file and then load that particular row and then throw out the rest of the memory. If you want to, like once you get it into NumPy or something, you will have to throw out the rest. So what we wanted to build was a specific file format where we store these matrices optimally so that we can randomly, like even if we want to go and fetch the last row, we can just go fetch it at constant time. And for that, what we essentially used is, by the way, this is not a entirely new problem. This has been thought of for a lot of years. If you just search for compressed parts row matrices, people have been using this for a long time. And what this essentially is, you have a matrix, and every alphabet that you see here is an interaction, and everywhere you don't see is we don't have any interaction, so that's a zero. And what we do is we get a row, and for that row we create a pointer here. So this row pointer 0 tells us that in here it starts at 0, and the second row starts at 3. So 3 is this particular index, and 4 is this particular index. If you don't have a pointer here, that means that row doesn't have any interaction with it. And what this allows us to do is this naturally allows us to index the data columns that we have with pointers. So what we do, we take these row pointers. We know exactly how much data is going to be in each of these columns because it's interactions, and it's all the interactions are stored as integers. So we know the exact number of bits that the integer will take in storage memory. And what we did is we sort of took these row pointers, we calculate the offsets that we have, and we put this into the header of the actual file format that we have. So what happens is we have memory, we have the file which will be around 100 gigabytes, which it will be in the sparse row format, And what we do is we put this offset in the row header. And what this offset will tell us is exactly where in memory to go and fetch how much of the data for a particular row. So here, if you think about it, if I want a specific row, let's say row 100, I'll read this entire header file into my memory. I'll go to the 100th offset. I know exactly where in the file system this 100th offset is. I go to that particular data point and fetch the entire row for that. This works really nicely because high C matrices are again symmetric and squared. So if we sum up all the rows, we can still get the column. So if I want to fetch a particular row and a particular column, I just need to fetch a row and then I just need to transpose it. That's it. So um, this was I we built this and we sort of used this for the purposes that we needed. But after we did this extension just became redundant because there's no other use case for this. So I just wanted to cover like lessons that we learned. If something else that exists for a specific task that you use, just use it. Don't duplicate it. It saves a lot of time. And if there's a very niche problem that actually deserves a new file format, then create a new file. I'll move on to the last part of my presentation, which is BRAD, which is the Bioinformatics Retrieval Augmented Digital Agent. So this was our lab is very interested in sort of digital twins and automating lab experiments in a way. So this was like a sort of stepping stone to what we were trying to do there. So the first thing that we wanted to do is we have the lab, we have the wet lab and the dry lab. We want to have an agent that can interact with these different parts of the lab and automate all of these tools that we do day to day. Like repetitive tasks can be automated by using AI agents. And what we went out to do is we built a AI agent that can with retrieve augmented generation. So retrieve augmented generation is sort of like normal LLMs that we train on a lot of data, but you also provide a source for the LLM to fetch from. For example, if we have a research paper that we want to that we have a specific question on, we can provide that to the LLM as a specific context, and whatever response that the LLM generates will be grounded on that particular piece of information that we gave it. And here we can see the general RAG overflow. Now, our agent that we built, we instead of just giving it access to files, for example, research papers and other things, we also gave it access to online databases and other software pipelines that the tool can run. So if you have a specific query, what the agent can do is it processes the query, it decides which tool to use for that particular use case. It goes forward, it knows how to connect to those tools and creates a response for that, decides if it needs to create documents for that, and then provides the response back to the user. And here, this is just like this is one of the parts of the tool where we go and fetch data from online services. The agent has access to Enrichar and other like literature databases like Archive, BioArchive, and everything. And here we'll use it. You give the instruction as a prompt to the AI agent, and the agent goes in, looks at all the databases that it has, and decides which tool to use. And yeah, this is like a sort of like a generic workflow of what we did. So you can plan, decide to run software, do online searches, document, and everything. So here, based on whatever input the user gives, you get outputs from the agent. And we want to like take this further. And over the past year, there have been other projects within the lab that have tried to automate experiments. For example, a person doesn't have to sit like 12 hours in the lab trying to do. We have a automation system that can perform experiments on the fly. And with that, I think it's time. So outside of this, I'm also extremely interested in once we get all of these parts working together, like we have dynamics, we know we can predict how cells move through, we can predict transcription factors to guide the cell towards a particular end goal. What I'm also interested in is instead of having to capture, like instead of having to capture, perform all these biochemical methods to infer data from, like capture data from cells, the only non-invasive way to capture data is live cell imaging. Like we can image the cell without having to kill it over time. And that if we can infer that from infer like higher resolution from these lower resolution images, can we actually predict other parts of the cell? For example, uh, in this confocal microscopy, you can actually look at euchromatin and heterochromatin parts of the cell. So can we look at how these two parts change over time just by doing light cell imaging is one thing that I'm interested in. And yeah, with that, thank you. If anyone has any questions. Yeah.
Thank you for that. And one question. So in the first project, if we have perturbation data, like CRISPR screen or drug perturbation data, can we improve your model, even validate your model or other applications if possible?
Yeah, that's a good question. So in our lab, we performed a perturbation of like the fibroblast to myotube perturbation we did with mmRNA. And yeah, for sure, we can use that data to sort of see how we can improve these. So right now, this A matrix that we get is purely from natural dynamics of the cell. So we can sort of see how the dynamics changes with perturbations, and we can definitely improve.
With perturbation, you taking?
This was with MyOD, and like we activated MyOD and suppressed fraction. Yeah. Yep.
Can go to slide number 18.
Yep.
Through a network structure. Yeah, this one.
Okay, this one. Yeah.
Yeah, so during networks, like only just adjacency matrices, right? So there's like no inhibition or activation between the nodes, right?
Yeah, that's correct.
Yeah, so I feel like between genes, there are many kind of patterns. So like one gene can activate the other gene, the other gene can inhibit, and there can also like form something like constellation structures.
Yeah.
So can you, do you think that cooperate with this kind of structure will help like improve this analysis and also like adds more meaningful?
Yeah, for sure. I think that's a extremely important question.
Yeah.
So here, for example, MyOD, we consider it as like activating all the genes, but that's not really true. So you're basically asking, can we incorporate these other structures?
Yes.
That's what we want to move towards. That's where these that's where this idea comes in, where for every gene and every transcription factor, we can sort of predict if it activates, if a transcription factor activates that gene or suppresses that, and then do that for all the transcription factors, all the genes. So you get a rich B prime matrix. Yeah.